We know that point mutations, single base changes that alter the sequence of amino acids in a protein, can alter that protein's structure or even truncate it, making it shorter than it's supposed to be. And we do know that there are cases where point mutations can have a dramatic effect on a protein's ability to function. But scientists weren't sure if point mutations explained all of the evolutionary innovations, the big changes in form and function that we see in the history of life on Earth. That changed with the realization that there was another type of mutation that could lead to not just an alteration of function, but to the gain of an entirely new function. The idea was first proposed in the 1930s, then formalized in the 1970s by Susumu Ono in his famous book. This idea relies on the fact that point mutations, single base mistakes when DNA is copied, aren't the only kind of mutations that DNA polymerase can cause. In addition to switching the correct base for an incorrect one, sometimes DNA polymerase can add an extra base called an insertion. Insertions can be small, like this extra C here, or they can be large, like inserting a whole string of bases, or even inserting a whole extra copy of a gene. When a whole second copy of a gene is inserted by mistake, it's called a gene duplication, and it's actually quite a common phenomena in prokaryotes and eukaryotes, and may actually even occur more often than normal point mutations. When a gene is duplicated, it becomes a lot easier for innovations to occur. Why? Redundancy. When you go from having one copy of a gene to having two copies, one copy can act like a backup, preserving the original function of the gene, while the other can accumulate point mutations, eventually making enough changes in the protein to lead to a new function. The point mutations are still random, and it's still possible that most of them don't necessarily go anywhere, or could even harm the backup copy. But with two copies, it's at least possible that some lucky individuals in a population will have one of their copies get enough mutations to become a new gene with a new potential function. Remember, this works because the original copy is the dependable backup, going about its normal function, while the other copy is free to accumulate all kinds of mutations, including those that might have destroyed the original function. Scientists think that gene duplication is responsible for many of the biological innovations throughout evolutionary time. So what does all this have to do with virology and with bacteriophages? So far, we've been talking about evolutionary innovations and what scientists already understood about them. This is the background information that set the stage for our project. In our research, we had an example of an evolutionary innovation we were trying to understand. This evolutionary innovation occurred in a bacteriophage called lambda. Normally, lambda infects its host, the bacteria E. coli, by recognizing and binding to a protein receptor on the cell's surface. This surface protein is a membrane pore called LAM-B. Once the virus binds to LAM-B, it injects its DNA through the pore and into the cell, starting the infection process where the host's machinery is taken over to make baby phage progeny, which then burst out of the cell. In laboratory evolution experiments, where scientists watch organisms evolve in the lab in real time, sometimes E. coli that are growing in the presence of lambda-phage will be lucky enough to get a random mutation which destroys the LAM-B protein, giving them immunity to the virus. Destroying a protein is pretty easy to achieve with a single point mutation, so we didn't really find this part of the evolution story too exciting. It's what's happened next that was really cool. Sometimes there were certain lambda-phage who acquired mutations that allowed them to bypass the missing LAM-B receptor and still infect the bacteria. How did they do this? By binding to a different protein on the surface called OMP-F and injecting its DNA through that pore instead. LAM-B and OMP-F have different shapes, so we thought a pretty radical restructuring of the phage protein that binds these receptors, called J, was required, making this one of those big change evolutionary innovations. So we wanted to find out how exactly this innovation occurred. Were a few point mutations enough, or was a gene duplication involved? What we found surprised us and represented a new way for proteins to succeed at multitasking. Somehow, our superphage was able to bind both the new receptor, OMP-F, and the old receptor, LAMB. We sequenced the genome of our superphage and found that the gene for the receptor binding protein, J, was not duplicated. So what was going on? How could the phage be capable of doing two things at once? It turned out that the phage had evolved a way to have non-genetic variation. This single phage genotype, the superphage that evolved, that was able to get around the host defenses and bind on BEF, is actually able to produce progeny phage with two different phenotypes, two different functions. How is this possible? Didn't I just tell you that proteins are bad at multitasking because a single DNA sequence is translated into a specific amino acid sequence, which folds into a specific protein structure? We think our superphage has a gene with a very special sequence. When it's translated into its specific amino acid sequence and the polypeptide is produced by the ribosome, instead of reliably folding into a particular defined structure every time it's translated, sometimes it folds into a structure that's a lot like the original version of the protein and combined LAMB, and sometimes it folds into a different structure, which is capable of binding the new receptor, OMP-F. So when the superphage infects a host and releases progeny, baby phage, some of them can only bind LAMB, and some of the progeny can also bind OMP-F. Because both versions come from the exact same gene sequence, we call this variation non-genetic. This non-genetic variation represents a new mechanism of evolutionary innovation. Instead of having to duplicate a gene, it's like two functions for the price of one. We suspect that for viruses, this could be a common way they are able to infect new hosts.